right? So to get started, amino acids are awesome precursors. We've actually seen some of these already. But we can use them to make hormones, coenzymes, nucleotides, porphyrins, antibiotics, neurotransmitters. The list goes on and on and on and on and on. Okay. I'm sure that is not exhaustive. So they're used for more than just proteins. I'm sure you can take this next slide and somehow morph it into a great faith-based devotional. I'm not the person to write that though. So, but that's because sort of like if I ever took the time to look up the scripture references of how God can use the lowliest beings, the lowliest positions to make the high and mighty. I mean, you can take like Christ was born in a manger in the lowliest position, and became ultimately the savior of the universe. Much like that, glycine, the lowliest of the amino acids, is used actually, and for us, for humans, to make the precursor for porphyrins. Okay, so what is a porphyrin?
It creates pores in the membrane.
No, it's a porin.
Oh, dang it.
So think back to last semester.
Oh, it's heme.
It's hemes. Heme, like hemoglobin, like big old heme that holds in hemoglobin, what does the heme do? It holds iron, right? And then iron then holds the oxygen and so on and so forth. So glycine, the smallest of things, can be used to make something so large and so intricate and necessary for our survival. Okay, and so in animals, glycine actually reacts with succinate. Where do we get succinate? We've heard of succinate. It's the citric acid cycle. And it forms delta aminolevulinate, which I'm sure that name will stick with you forever. Now in plants and bacteria, it's actually glutamate instead of glycine. Why? And this is where you really want to take the evolutionary biologist and say, explain this. Why do we use glycine, something more simple, and plants use something okay. And the iron, actually, I just want to let you know, until after you make protoporphyrins, not inserted early on in there. That's just a little side note. What happened there? There. Okay. I'm like, something. So the top is the animals, plants, bacteria, and the bottom. The delta aminolevulinate is the common metabolite. So if we just look here, I've given you the enzymes on this already. I didn't cover them up. It should make sense. Notice, what does the box indicate? Just a review from Gen Chem even. It's an intermediate. I know sometimes people forget, and when we have that box, it means it's the same enzyme. So we just have a simple enzyme. We take succinyl-CoA, glycine, we make delta aminolevulinate. It's a synthase. Not difficult. Plants is a little. One thing I like to show about the plants is the fact that it uses something that we haven't seen used in metabolism before, and that's tRNA, which is really odd. But it takes glutamate, and it takes the glutamyl tRNA synthetase to make. This is, by the way, this is the same thing that would be used at the ribosome, to go to the ribosome to make proteins, when we charge it with the amino acid. But then what plants do is they use a reductase. The tRNA is just a great leaving group. Okay, so it kicks the tRNA off, and it makes it glutamate, semialdehyde, and then there's this mutase that goes on to make delta aminolevulinate. All right, so now we made the delta aminolevulinate. Please do not memorize all of these enzymes. I would probably expect you to recognize what class of enzyme, that kind of thing. Well, the reason why is there's just lots of steps. So once again, you could take almost a whole, at least one lecture, if not more, and talk about there are diseases associated with all this. There are just some points that I wanted to make. One, once again, we're starting with the delta aminolevulinate. the ultimate goal is to make heme. These are the enzyme names. So you can see where you have synthases. We have a decarboxylase. But the very last one is the iron, is when the iron gets incorporated. And that's using a chelatase, which means it chelates the iron, the ferrochelatase. Okay. So iron is not incorporated to the very last step. All right. Porphyrin biosynthesis is regulated by the concentration of heme, which that makes sense. It's feedback inhibition to early on. There are genetic defects all along the way. You can, anytime you have so many enzymes involved, you can imagine any point in the process, if you have a faulty enzyme, like a faulty genetic mutation, you can alter. Even just altering the ratios can cause a phenotype. I don't want to say causes something bad, because just because someone has a disease doesn't make him or her a bad person, but it can cause a phenotype. All right. So these diseases many times are called porphyria diseases. Okay, so King George III, by the way, which is for those who are history aficionados, I was going to say Nets, aficionados, he's the king, the British king during the American Revolution. He suffered from a porphyria disease. Caused abdominal pain, it caused neurological problems, neurological disorders. They had problems with iron metabolism. There's another form of porphyria. This one is it's rare, but it does result in an accumulation of one of the precursors. Okay, and once again, I wouldn't ask you which one is it. That's your porphyrinogen one. It causes your urine to turn red. Your teeth will actually glow. Okay, I had one of my students in Michigan. I don't know if she had this disease. I really wanted to ask. But I used to pass out these little index cards. This is back this many years ago. Pass out index cards, I had all these students. I'm like, put your name down, like if you go by something else, and tell me something about you to help me remember who you are. And she told me she was my teeth glow. I'm like, I wonder if she's got porphyria. But she's like, I'm glass at a party. And I was like, okay, yeah, okay, thanks. But yeah, they're also really sensitive to sunlight. They have problems with their skin. They have severe anemia, so they turn very, 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 very white or pale. And so, does anyone know what that phenotype looks like? Results in? So it's the belief it's a cause of vampirism. So they actually have certain porphyrias where they crave blood, and so they want to eat or drink blood. They may want to eat raw meats, but yes, it is believed to be like the historical and genetic basis of the stories of our vampires. They do not turn into bats, though. And they don't all speak with Romanian accents. All right. Are there any questions? We have the creatine and phosphocreatine, and how we get that from glycine, arginine, and methionine. And I say methionine because it's really Adomet or sand that does it, but technically, you already know that sand comes from methionine. We've gone over that time too. Phosphocreatine is a very important buffer, especially for your muscle cells. And it should go without saying that it comes from creatine. Okay. So here we go. Make it a little bit smaller. So you see we have glycine and arginine. This first enzyme in this pathway will yield guanidino acetate and ornithine. What can ornithine go on and do? Urea cycle. Or in theory, it could also go on to make more arginine if you needed. But yes, urea cycle. Mm-hmm. What class or classes of enzymes because like what's really happening here? I can understand two classes of enzymes being used here. Transferase, and this is a transferase. And as I say, some people would say, oh, it's a synthase, so we're taking two things and building, but yes, no, this one's a transferase. What is being transferred?
Glycine.
Pardon?
Glycine.
I know that'd be so easy. Too bad, it's not. You know, say, oh, it's a glycol transferase. What is, does anyone remember what that, it's not an imine. And it's a functional group that, I don't know if I've ever, I think I'll only say it during this lecture. But it is that functional, it's an amidino group, okay? So it's an amidino transferase, and that way you can see what it is. Which I guess kind of looks like an amide and an imine put together, amidino. It's not something that you use a lot. This is one nitrogen short of being guanidine. But the guanidino acetate name should make sense because remember guanidinyl groups? That was one of the functional groups on Baird, was that chapter 1 from last semester. It's a functional group that makes arginine. And so acetate is acetate. You see that as the acetyl group. All right, the next enzyme is where methionine was used to make Adomet. And does anyone remember what does the ADOHCY stand for? HCY was the 3-letter for homocysteine. So it's adenylylated homocysteine. Unfortunately, it's not adohistine. I don't. Everyone says Adomet. I don't think that they ever. It doesn't have a cool name like that. SAW, SAH. Sometimes you see it abbreviated as SAH instead of SAM. Okie dokie. And it makes creatine. So what class of enzymes is this one? What does Adomet do? I mean, it just does the same type of reaction over and over and over again. There's a class of enzymes that's what they use it primarily. It's a methyltransferase, and that's all you need to know. It's methyltransferase. You're methylating guanidino acetate to make creatine. And then last but not least, we have creatine becoming phosphocreatine at the expense of an ATP. So what class or classes of enzymes could this one be? It's a kinase. What's this one? I do want you to know the specific name because it's an important enzyme. It's creatine kinase. It's used. It's also used for good bio level marker too for like muscular development. And also for like cardiac issues. Okay. Are there any questions over this one? It's not, other than trying to remember what an amidino group looks like. It's pretty straightforward. The next biomolecule that we're going to talk about is glutathione. We first mentioned glutathione last semester in chapter 3 or 4, I think. Do you remember what is glutathione? I would almost get bonus points right now if you knew.
Amino acid.
It is. In what way? What is it made up of?
It has a sulfur group by diamine bar.
Yes, it does have a sulfur group. Where have we seen sulfurs from? What? Methionine, cysteine. So those are already some of the good things. Glutathione is a tripeptide. They abbreviate it a lot of times as GSH, and then whenever it forms a disulfide to itself, it's called GSSG. It's a tripeptide. It's also a buffer. Okay, for especially we talked about the first time that we talked about it was the fact that it was for whenever proteins were first starting to fold and form. So you can imagine the protein's got cysteine. Cysteines can easily form disulfide bonds, but you don't want every cysteine to form, umpteen, various possible disulfide bonds. So glutathione was used as a buffer in that sense because it's really tiny, it's relatively tiny, it has a cysteine in it, and so it just forms a brief disulfide bond, and then it's easily reversible. Okay. and it's ubiquitous. So it helps with protein folding. It does help regulate a buffer of thiols. So in that sense. And so it does reduction and oxidation with respect to thiols. Oh, right. So, what's so important about sulfur? We've already talked about disulfides with protein bonds, protein structure. But it acts like a what? What?
Iron sulfur citrate.
It can be used for iron sulfur citrate. Mm-hmm. So that means it obviously has to play a role in redox reactions. What else does it do? It's a great nucleophile, and it's also a great leaving group. So there's lots of things that sulfurs can do. Okay, so, but yes, it is a redox buffer in that sense. All the maintenance of the thiols and disulfides, and then it helps with the iron oxidation state, especially for hemes, but also for iron sulfur protein. I don't know why I didn't show I apologize, I didn't show it in the reduced form. I only showed it oxidized. But that's easy. To draw the reduced form, whoops. Gamma. That's a C, not an L. There it is. Okay, so that's reduced. Reduced means there would be two of them if we're going to do a balanced equation, and then it would have a thiol. Oxidized is when you have the glutathione, the GSSG form, the disulfide form. What does that gamma mean? What is the importance of the gamma? It's what? It is. So, yes, it's the locant for it. Remember, it's alpha, beta, gamma, delta, all that kind of stuff for biochemistry. This is just a two-step process, not anything major. Okay. Let me make this a little bit smaller. So I'm actually going to point out while you're looking at this, the gamma had to do, because if you did glutamate, how many carboxylic acids does it have? For glutamate, carboxylates, I should say. Two, right? It's got the regular alpha amino one, and then it's got the one in the gamma position. So that's why, in theory, all this is a tripeptide. These are all peptide bond formations, like what we've seen from last semester, except for the glutamate. Instead of it being the quote-unquote normal one that's making the peptide bond, it's the terminal one, the one that's on its R group. That's why they call it gamma glutamyl. That's why its full name would be gamma glutamyl cystinyl glycine. Of course, no one's going to call it that. It's okay. So in the beginning, for the first step, we have glutamate and cysteine coming together using an ATP to make gamma glutamyl cysteine, making the dipeptide. So what class or classes of enzymes can this be? It's a synthetase. So if you were to name this specific synthetase, what would you name it? Gamma? Glutamyl? Remember, it's got the YL instead of the main, or I should say the main part. Gamma glutamyl, cysteine synthetase. Exactly. And what's the whole purpose of ATP once again? Once I just made a new word. Once again, both these steps here. It's just a review from why use ATP? Why do we use ATP?
Activates the
It activates. It's great. It activates the intermediate that's formed. It's a great leaving group in the form of ADP and phosphate. So that's what it does. Okay. And then the second step, we have glycine coming in, without cheating ahead or anything like that. It makes sense. Classic enzymes make sense. It's a synthetase. What synthetase is it? What are we synthetizing, so to speak? It's glutathione, so this is glutathione synthetase. Yep. Okay. Are there any questions over glutathione? Like I said, the only thing to make it reduce is you have two glutathiones that come together and make a disulfide bond. Alrighty. So next up, we're going to look at the neurotransmitters, which is actually a nice segue to the next chapter. The last chapter that we're going to be doing. I do want you to know what the neurotransmitters do. You need to know the important enzymes and the cofactors. You're going to see there's a similar step pretty much in all these cases, and so it's not hard in that sense. You learn one, kind of see the same thing over and over and over again. Okay. Once again, try not to look ahead. You've already looked up the notes. Just because I want you to know the logic rather than just reading off the notes. We're going to do dopamine, norepinephrine, and epinephrine. What are the common names or other common names for norepinephrine and epinephrine? What is epinephrine? It's adrenaline, and norepinephrine is? This is a logic question. Epinephrine is to adrenaline, what norepinephrine is to people. What's the only difference between epinephrine and norepinephrine? It's noradrenaline. Like I mean, literally, it's
If you could read my mind, you would hear the answers to that more than not. You don't mind.
You gotta get over that fear. You gotta get over that fear. Just like witnessing to somebody. Okay, so first up we're gonna do dopamine. Dopamine comes from tyrosine. Okay. And actually I'm just gonna go through the spiel first and then right. I would expect, at this point in time, you should have an idea what dopa and dopamine are, in the sense that it's involved in Parkinsonism, Parkinsonism, of course, is the generic term for the phenotype. Parkinson's disease is a very specific disorder. And then dystonias, what are dystonias? What is dystonia? What does that term mean? Technically, dystonia is a phenotype. It's what?
Away from normal morphology, right?
But what's dystonia? Nick? Yeah, so people that are so starting what I had, so dystonia, idiopathic personal dystonia, must be learned. So dystonia means these are people who they don't control, it's like the opposite of being paralyzed. These are people who cannot control the muscle movements, muscle control, so they usually have hypertone, and that kind of thing. Okay, muscle spasms. All right. So, just as a little side note, the treatment usually, many times, can include L-DOPA, but there's a whole brain barrier issue. Like if you know anyone who has take DOPA, you have to take really, really high concentrations of it just to get a small fraction to cross over. Is that holy holies? And then like the story that I had, it was called DOPA non-responsive. It didn't matter. We can get into carbidopa versus levodopa. We can talk about that later. Okay. And in the overproduction, there's been lots of different disorders, neurological and psychological disorders associated with it. Tetrahydrobiopterin, I don't think that we covered tetrahydrobiopterin yet. It may have been on that list of cofactors from like last semester. We don't see it a lot. I don't, and this is the problem that I've seen a lot of people get confused, is between tetrahydrobiopterin and tetrahydrofolate. That's the H4F or H4 folate that we've seen. Remember, tetrahydrofolate, that single carbon transfers. Tetrahydrobiopterin is a redox reactant. So, a redox reaction, I should say. Tetrahydrobiopterin becomes dihydrobiopterin. And then we have, if you notice, O2. Yeah, you guys right. O2 becomes water. So the only difference between tyrosine and DOPA is what?
Alcohol?
An alcohol. So what class or classes of enzymes can this be? I can think of two possibilities here. A what?
Hydroxyl transferase.
A hydroxyl transferase, which they commonly call a hydroxylase. Mm-hmm. And then technically, and then, by the way, this one is a hydroxylase, but technically, what big class of enzymes is this that we've already talked about? Like hydroxylase technically is a little subclass, but what's the big class? It's not an oxidase, it's oxygenase, right? Okay, and technically this one would be a monooxygenase versus the dioxide. But yes, it is a hydroxylase. And so if you're going to name it, what would we name this specific hydroxylase? What is it hydroxylating? Yep, that's it, tyrosine hydroxylase. If you had called it tyrosine monooxygenase, I would give it to you as well, because technically it is a monooxygenase. And then the pink step is important because it's a common step you see in a lot of these neurotransmitters. We're using our good friend PLP, pyridoxal phosphate. Okay. We were removing the carboxylate group, I guess you could say, in the form of CO2. So that's the only difference between DOPA and dopamine. So we got rid of the carboxylic acid portion of it. So what class or classes of enzymes could this one be? It's on the last exam. On the bonus. Remember? PLP does 3 types of reactions. Racemization, aminotransferases, and decarboxylation. This is decarboxylation, so what would this enzyme be? Yeah, don't overthink it's a decarboxylase. Okay? And if you're going to name it, you would name it dopa decarboxylase. However, don't write that down. Because this enzyme is very promiscuous. So what kind of compound is this right here? It's aromatic. It's an aromatic amino acid, okay? And so that's why I just wanted to put that out. This is aromatic amino acid decarboxylase. This enzyme gets around.
Could it be inhibited by other aromatic ring structures? Or?
It could, in theory. Or it could take other aromatic amino acids and remove the decarboxyl groups. Remove the carboxylate groups. Okay. So next, we still haven't made norepinephrine and epinephrine, but you make dopamine first in order to get it. Both of them, by the way, are involved in product life. And they bind to the adrenergic since it's adrenaline. So the first step is you take dopamine, we're turning it into norepinephrine or noradrenaline. I'm not going to ask that question again. Okay, notice we use the ascorbate to become dehydroascorbate. What is ascorbate? This is one that I don't know that we've had another example of ascorbate before. What is ascorbate? Well, what is the difference between ascorbic acid and ascorbate? A hydrogen. One of them is the carboxylic acid form, one's the carboxylate form. Mm-hmm. So what is ascorbate or ascorbic acid? I should hope you would know this. You guys should know this by this point in time. Vitamin C, thank you. This is vitamin C. That's the official name.
Oh.
Just like alpha tocopherolic acid would be vitamin E. Alpha tocopherolic.
I didn't ever.
Retinoic acid is vitamin A. So you did not in A and P, you guys don't have to know what ascorbic acid is. Don't go nickel scrolling like that, okay? That's what you should know. All right. We have failed you. We have failed you. We have failed you. The ascorbate is vitamin C, or ascorbic acid.
I wish we had a nutrition course. Yeah.
I mean, I would have thought that was covered in biology,
No.
A and P. So, yes. All right. But let's focus. Focus, focus. Let's put our blinders on. We're horses. Now, we have dopamine. What's the only difference between dopamine and norepinephrine? It's an extra alcohol group. So what class or classes of enzymes can this be? It's a hydroxylase. If you were going to name this enzyme, what would you name it? It is a dopamine hydroxylase. Now let's up it. What position is being hydroxylated? Is it alpha? It's beta. Because alpha means right next door. So if you look at the amino, that's why we have alpha amino acids, because it's the carboxylic acid is alpha to the amino group. So this one's a beta. And so that's why you put it all together. It makes sense. It's dopamine beta hydroxylase. And this specifically tells you that what position gets hydroxylated versus the other hydroxylase, which is on the phenol. Okay, all right, I mean, on the benzyl ring. All right, and then we have good old Adomet coming in to go from norepinephrine to epinephrine. So what class or classes of enzymes is this one? Adomet, methyltransferase. Now this one is a little trickier. This one's got a very, very, very specific name. First of all, so let's break it down. And this is a methyltransferase. What position is being methyltransferred, being methylated? The N. Okay, so that would put it together. This would be what kind of methyltransferase? An N methyltransferase, because the N is a locant. Remember, technically it's italicized. This is one time where you'd say, oh, it's norepinephrine N methyltransferase. That, and I don't know why, but they actually do go based off of the real organic chemistry name. So instead of the common name norepinephrine, that's why I just want to point that out. Because it's phenyl ethanolamine N methyltransferase. But norepinephrine is so easy to say versus phenyl ethanolamine, but that's why that is. Do you see why it's this is ethanol amine, that's the phenyl group. Okay. So that's how we get adrenaline, essentially. All right. Next up is good old GABA. Which, if you went to Brenda Lee's talk, she talked about GABA. GABA is a common name for gamma aminobutyrate. Butyrate, what does butyrate mean? You probably didn't learn that one in organic chemistry. You probably called it something else in organic chemistry. What's the but? 4. Right, normally you would say butanoate, right? Because you took butanoate, most or traditional organic chemistry books will say, you name the carboxylic acid, butanoic acid, when you lose it, becomes carboxylate, you drop the acid part, you add 8, so that's why it becomes butanoate. So that's why if you called it gamma aminobutanoate, people would also, first of all, it still fits, it's still gamma. Butyrate is because the old name for butanoic acid was butyric acid, so that's why it's It is an inhibitory neurotransmitter. It's linked to epilepsy. It's also linked to things like, you know, anxiety disorders, depression. There's lots of different things. Alright. So, what amino acid forms GABA? It's glutamate. So what enzyme is that one right there? It takes PLP, removes the CO2 to make GABA. It's a one-step process. It's a what? It's a decarboxylase. And what would you name this specific decarboxylase? What? It is glutamate decarboxylase. We know it's not aromatic amino acid because it's not an aromatic amino acid. So yes, this is glutamate decarboxylase. Yep.
Why is the box pink for this one? Is that also?
It's the same step.
Okay.
If you notice the last step for all of the last step is the same. To make dopamine, to make GABA, okay, to make the next one. The next one is histamine. That's why I tried to show the histamine. I truly hate thee right now. That's a Shakespeare rendition. That's the reason why I'm carrying out toilet paper today. Not because I have to go to the bathroom, but because I keep blowing my nose and constantly snotty. So, I would really, really like to take an antihistamine. Give me a Benadryl or two and let me go sleep it off, but unfortunately I can't. So, histamine, of course, is power vasodilator. It goes out of whack when you get the, stuffy nose, not stuffy, like the snotty, and well, some people get stuffy noses, the eyes, just a pain. It's also linked to stomach acid. And of course, the antagonist that you use is called an antihistamine. All right, so what amino acid makes histamine? Stop. It's histidine. I've actually seen people accidentally get confused and call it the wrong thing. But it's histidine. And so what specific enzyme is going to be making this one? Oh, see, you guys are batting 1,000. It's histidine decarboxylase. I told you, it's not hard once you get in the hang of things. And finally, one of our favorites, at least right now, I'm sure all of you right now are thinking it. Not thinking it, but that's serotonin. We'd like to have serotonin go to sleep. Also, it's involved with sexual arousal and ejaculation, sexual dysfunction problems, possible link to homosexuality. Sounds a little questionable. I don't remember if that link actually still
It works.
It does. Isn't that awesome? Okay, so yes. Little side note, I was at a meeting, at an international meeting, I don't remember, I think it was the one that was in Banff, many years ago, and there the guy that was speaking, he was going over Drosophila, fruit fly, Drosophila melanogaster, and they were looking at, this was an International RNA Society, so they were looking at the microRNAs that's involved with turning off some of these genes. And whenever he did it, he was talking about how that they had these metal fruit flies, and he's like, we use this microRNA, and this was a relatively new field, and it caused, a change in sexual preference. And honest to God, it was really quiet, hundreds and hundreds of people there, and somebody said, found the sex gene, or whatever, and of course they just lost it. No, they found the gay gene, and of course they just lost it, and I felt so sorry for the first few years, so, but yes. Serotonin syndrome is something that's really, yeah.
So if I understand this correctly, they're essentially saying that eating turkey can make you gay.
No. Well, that's a different type. That's a different type of gayness. Yes, that's guillotine. No, I mean, you would have to, in this theory, you would have to have an underlying defect, and you'd have to have so much turkey that you're probably going to die from something else. Okay, but no. Serotonin syndrome, by the way, is hyperserotoninemia. It's actually highly fatal. It's usually from taking drugs that combat against each other. So that's why if you're on SSRIs, what are SSRIs? What's one of the S's stand for in SSRI? Serotonin. Okay, does anyone know what the other letters stand for? Selective serotonin reuptake inhibitor. And so what it does, is, it allows serotonin to be around a lot longer for people to combat depression in particular. Usually, some types of anxieties also are linked to SSRIs as well. Okay. All right. So what? Oh, I didn't realize it's cut it off. What is the precursor? What amino acid does serotonin? Is what? Tryptophan. Good old turkey. And that's kind of a little misnomer, by the way. That people say, "Oh, you eat turkey for the tryptophan." Turkey does have, but so does chicken, and so does beef. So does, lots of things. It's not like it's. Yeah. All right. Once again, we have tetrahydrobiopterin becoming dihydrobiopterin. So what's the only difference between tryptophan and this is sometimes abbreviated as 5-HT for 5-hydroxytryptophan. So what's the only difference? Don't overthink it. It's an alcohol. It's a hydroxy. That's why it's called 5-hydroxytryptophan. Okay. And so, but once again, a lot of times if you see this in literature, it's even abbreviated as 5-HT. And so, what class of enzymes can that be? That first step. Knowing what you know so far, and you've seen that over and over again. Is a hydroxylase, and specifically, what hydroxylase is this one? What? It's tryptophan hydroxylase. And then the only difference between 5-HT and serotonin, which technically speaking, I know some people say 5-HT is the same thing as serotonin. It's not. It's the precursor. It's the direct precursor for it, but it's just that for most people, once you have 5-HT, if you increase 5-HT, you can increase serotonin levels, or decrease, the drug related. What it uses PLP, it's removing a CO2 group, so what class of enzymes is this? It's a decarboxylase. What decarboxylase is this one? What? Ooh, yeah, 5-hydroxy, blah, blah, blah. Or our good friend, the loose one, the one that gets around, is aromatic amino acid decarboxylase. Tryptophan, once again, tryptophan, tyrosine, phenylalanine, those are all the aromatic. Well, technically histidine too, but those other three have the traditional, benzene rings. So it's the aromatic amino acid decarboxylase. Are there any questions about any of this? Anything at all?
What was the SSRIs involved with?
What do you mean?
Was that just a fun factor with us?
Well, I would expect you to understand, like, just knowing, here to come up with ideas of what they were used for. I don't remember. There used to be a suggested problem that talked about SSRIs, but I don't know if it's still in the most recent edition of the book or not. But it's selective serotonin reuptake inhibitor. So then just break it down. So SSRI, selective, I don't remember, it's selective, serotonin, inhibitors, okay? So, if you take an SSRI, in theory, will your serotonin concentration go up or go down? It's going to go up. Because the reuptake, so don't hate me for my beautiful drawings. Of course, the idea is serotonin ultimately gets released For the presynaptic neuron, it would be taken. It would go in. I can't think. I'm tired. But let's say that those are the receptors to take them in, and then the SSRIs bind to those or bind to specific ones, and that way more serotonin stays out longer, in theory. Okay. So, you could then talk about some of the side effects. Oh, it's going to be drowsy. That kind of stuff. Or I would not. The link to, homosexuality is very controversial, so I wouldn't be saying, I wouldn't be asking you, on the bisexuality spectrum scale, what would you expect? That would be ridiculous. Okay. Any other questions? Oh.